Page 57. And the first lab we did in this new unit is on enzymes. And so it says, for number 1, identify the reactants, products, and enzyme in a chemical reaction. So I went ahead and wrote up here a chemical reaction. And it is the chemical reaction that we used when we did this lab. You don't even have to know the names of these things. You can still figure out who the reactant is and who the product is. And then I want you to know who the enzyme is. So which one of these up here is the reactant?
The one on the left.
The one on the left, H2O2. I'm just going to put an R there. And that is hydrogen peroxide, in case you recognize it. That's hydrogen peroxide. And then who's the product?
H2O plus O2.
So we have 2 of them, H2O and oxygen, which is a gas. And then who's the enzyme?
Catalase.
Catalase. I just put a lowercase e for enzyme because uppercase E we use for energy. So I used a lowercase e for enzyme. Ready okay on that? Simple enough. Okay, great. Okay, number 2, you're supposed to be able to define these things. And what I really, really want you to also be able to do is be able to identify them in a picture. And so I recall this was something that was on our test last week. You recognize this? Yeah, it was on our test last week. And so, or in your lab manual, page 57, a similar type of piece of artwork, page 57. So the idea is, would you be able to label this picture if I took all the words off? Does everybody see how I scratched out the words? I guess I'd scratch that out too. Scratch out everything. And would you be able to label the picture? And I could tell last week on your test, most everybody could label the picture. Let me just make a quick comment. The only time I see people getting a little confused is being able to tell an enzyme from a substrate. So everybody take a look at the picture. Does the enzyme change in the chemical reaction? No. Does the substrate change in the chemical reaction? Yes. What does the substrate become? Nope, the substrate does not become the enzyme. It becomes products. Thank you. It becomes products. It was too easy an answer, but people mess it up. Products. The substrate is simply a reactant, and of course it becomes products. And that's the only time I see people getting messed up. So please remember, enzymes stay the same throughout the whole process, but substrates become products. So you can see them changing into something else.
Ending product.
Sir?
Ending product, like out of product line, like a factory.
Exactly. So the substrate would be the raw material going in, and then what comes out the other end that you sell would be the ending product. That's very good, Louie. Okay, so I added this little question. No, I didn't. What is the effect of an enzyme on the activation energy of a chemical reaction? This is still part of number 2. So I did a quick graph. What is this little E with a letter A on it? Does anybody know what that means?
Rise in energy.
Energy of activation. So what is the effect of an enzyme on the energy of activation of a chemical reaction? Well, first of all, I have a yellow line and a blue line. Can you see that? Yeah. One of these lines is with an enzyme, and one of them is not. Which line would be the line with the enzyme?
Lower one.
The lower one. And so what does an enzyme do to the energy of activation? It lowers it. So I'll write that down. Lowers the energy of activation. An enzyme will lower the energy of activation. Thus, an enzyme will make the chemical reaction go faster. Ready okay on number 2? I just threw that graph up there, but it's simply a graph from your textbook or lab manual. Okay. Question 3. What happens when an enzyme is denatured? What does denatured mean? It changes shape. Good, Michaela. It changes shape. Okay, is that a problem?
Yes.
It's a big problem. The enzyme will stop working if it changes shape. Okay, what conditions might happen, what cause this to happen?
High temps.
High temps, great. Anything else? Extreme pH. That's good, Celeste. Extreme pH, like super high pHs or really low pHs. Okay, number 4. What's the function of catalase? Check it out, page 58. About 2/3 down the page on page 58, you maybe already boxed this chemical reaction. What is the function of catalase?
It's an enzyme.
It's an enzyme, and what's its function? So it makes water and oxygen from what? Okay, so it speeds, I'm going to combine your answers. Speeds the breakdown of, that's supposed to be a W, of hydrogen peroxide. Guys, H2O2 is hydrogen peroxide. So hydrogen peroxide is breaking down there, and catalase makes it go faster. How did we know if catalase was working?
Bubbles.
Let's see the identity of the bubbles. Oxygen, thank you. So we looked for bubbles. I think we saw a lot of bubbles. We gave them 3 pluses, 2 pluses for okay, 1 plus if just there's a few. No bubbles, we gave it a 0. Okay, what effects do different temperatures have on catalase activity? Well, let's check out page 60 and look at our actual data. Page 60, table 5.2. Okay, how did catalase do in test tube number 1, which I think was the refrigerator? How did it do there? Did you get some good bubbly? Yes. Did great. How about test tube number 2, which was the warm water bath? Still did great. How about test tube number 3, which was the 90-degree water bath? No. So then the question was why? It looked to me like it was working pretty good at most of the temperatures, but we got it super hot and it stopped working. Why? It denatured. So just as Louis had mentioned earlier, high temperatures are bad for enzymes. It denatures them. And our data support that. Oh, number 6 is an omit. We did not do the activity for number 6, so it is an omit. Number 7. What is the effect of a pH outside the optimum range of an enzyme? Our data is on page 62. Okay, test tube number 1. I think our data indicate test tube number 1 had a pH of about 1. Yeah. Test tube number 2 had a pH around 6 or 7. And test tube number 3 had a pH of around 14. Well, for me, test tube number 1 and test tube number 3 look like extreme pHs. What Celeste was mentioning earlier, extreme pHs at one end of the pH scale or the other. And looking at your data, what is the effect of pH outside the optimum range? What does that typically do to an enzyme? Denatures it. So for the next part, I actually drew some test tubes there. And these test tubes are supposed to be test tube 1 from page 62, Test tube 2 from page 62 and test tube 3 from page 62. Test tube 1 had acid in it, test tube 2 had water in it, and test tube 3 had sodium hydroxide. Now just above each test tube, I'm going to go ahead and write down what the pH was. I think it was a 1 for test tube 1. Did we get 6 or 7 on test tube 2? Yeah, so 6 to 7, that's what I thought. And then the last one, what did we get? 14. Okay, did we get bubbling in test tube 1? No. Test tube 2? Yes. Test tube 3? We got bubbling test tube 3?
No, it was like a 1. So like no.
Just a little bit? A bit. Okay, can you figure out what might be the optimum optimum pH for catalase from this data?
Yes.
What? Yeah, near neutral. Work best near neutral. And when you went outside the optimum, too low or too high, it didn't work very well. So I'm just going to back up to that question. What would acid do to catalase? HCl is acid. Denature it. And what would base, NaOH, do to catalase?
Same thing.
Denature it. Okay. Morning, Colton. Here's a review sheet. Okay, so that's exercise 5 on enzymes. Do we need to repeat anything? Sure. Okay, so guys, I'm going to move to exercise 6 on photosynthesis, and if you'll join me, we'll be on page 67 now. Pretty much goes without saying you're responsible for that chemical reaction that's on page 67. I bet you've already boxed it or highlighted it on page 67. That's the summary of photosynthesis. So what are the reactants of photosynthesis? What are the products of photosynthesis? Carbohydrate. Who told me that? Good job, Jennifer. Carbohydrate, which is CH2O. That's carbohydrate. And what else? Oxygen. Yeah, that's CH2O. That's carbohydrate. CH2O is carbohydrate. Okay, you also need some light. And I just want to comment, one more thing that's not on here that you know that autotroph needs is some specific pigment.
Chlorophyll A.
Chlorophyll A. Good job. Chlorophyll A. You got to have chlorophyll A. Everybody okay on number 1, the reaction? Okay. Still on number 1. In what organelle does photosynthesis occur? Chloroplasts. Thylakoids and stroma of a chloroplast. Very good. And guys, there is a pretty decent picture on page 67 of the chloroplast. Similar picture was on our test last week. Most of you were able to successfully answer those questions on the chloroplast. Well done. Okay, page 68, paper chromatography. Page 68. So this is the day we mashed the leaf at least 10 times. Some of you mashed it so well, had some excellent results on this because of your persistence in mashing. Good job. So you got really deep pigmentation on your paper. And you may remember we had to pour a solvent down into the bottom of the test tube, and that solvent would rise on the paper, separating the pigments for us. So you may remember what kind of solvent we used. It was acetone. It's like in fingernail, yeah. And I want to remind you that it wasn't water. Because it wasn't water, a few of you attempted using a dropper pipette, and you were a little frustrated because it would run right out of the dropper pipette. And that just goes to show you that because it had no charge to it, it wasn't adhesive to the dropper pipette like water. Water's got some charge, so it'll stick to stuff, but not that solvent. It would just run right out of the dropper pipette. It was hard to control. But it was a nonpolar solvent. Okay, looking at the sentence that you may have written on page 68, I'd like to answer the next question. How does paper chromatography work?
Separates pigment based on solubility.
Good. So it separates the pigments that we mashed on the leaf based on the different solubilities of the pigments, how well they dissolve. And again, just because of experience, I knew not to use water because I knew water wasn't going to dissolve those pigments, just based on experience. So I picked a nonpolar solvent like acetone. Okay, so I drew a chromatogram up here, and it kind of represents what we had. There was yellow at the way top, and then there was a space, and there was some more yellow, and then come on down, there was some green, and then right behind that was some more green. Now, you're going to have to know which pigment was which on my pretend chromatogram. So let's start with the top pigment, which was the yellow one way at the top. What's the name of that? Carotene. And this is off of page 68. What's the next one? That's correct. The third one? Chlorophyll A, our essential pigment. And the last? Good. And then I threw in this little question, which one is the most nonpolar? Carotene. Carotene. How come carotene is the most nonpolar? It went the furthest. It dissolved the best in the nonpolar solvent, and it went the furthest. Okay, which was the least nonpolar? Chlorophyll B was the least nonpolar. It didn't dissolve very well, and it didn't move very far. So if it moved far, it was the most nonpolar. If it didn't move far, it was the least nonpolar. But okay on that one. Okay, question number 3. Study the absorption spectrum of chlorophyll, page 72. On your way to page 72, can I express my admiration one more time of page 70? It's quite lovely. What's so great about page 70?
It shows the spectrum of light.
Shows white light is actually made out of all the colors of light. That's great. Okay, now page 72, we got this graph. Well, let me also tell you this. Some of you didn't get a similar question that I was inspired by this page to put a question on your test last week. You guys remember I had this graph, and it had the colors of light across the x-axis and absorption on the y-axis, and then it had a line, and you had to tell me what color the pigment was. Some of you didn't get it. So I want you to take a look at chlorophyll A. How can you look at this graph and know chlorophyll A is a green color?
Because it absorbs the least light at green.
Yeah, it's not absorbing in green. Or I can look at this graph and tell you that the carotenoids are going to be yellow, orange, and red. Because what's happening for the carotenoids in yellow, orange, and red? They're not absorbing. So whatever color you don't absorb, you reflect that color, and that's what we see. We see what you reflect, not what you absorb. Can I just mention that because we had a little bit of difficulty with that last week?
I think you brought up when we went over that before about plant life on the ocean floor. It doesn't receive sunlight, although it's still colored. We can't see the color. All it's blue.
It does.
Right, until you shine a light on it.
Until you shine a light on it, and then there's the color. That is true. Okay, number 3. What colors of light does chlorophyll absorb the best? Let's do chlorophyll A because it is the primary pigment of photosynthesis. Look at chlorophyll A. What colors of light does chlorophyll A absorb the best?
Purple and blue.
Okay. Anything else? I think we got a pretty strong orange in there, huh? Right over there on the right-hand side. Got a nice peak there. What colors of light does chlorophyll absorb the least? Green and yellow. Okay, then how did you know? Did you look for absorption? Did you, to know what it was absorbing the best, did you look for peaks or valleys? Peaks. Yeah, peaks tell us what it's absorbing the best. Valleys tell us what it won't absorb very well. And that just makes sense because the y-axis is absorption. So to figure out what it's absorbing, you look for the peaks. To figure out what it's not absorbing, you look for the valleys. So really it's just asking how do you properly read the graph. But okay on using that graph.
Yes.
Okay. So we have learned that plants have more than just chlorophyll in their leaves. Other pigments are there. So question 4, why is it beneficial to have several plant pigments involved in photosynthesis? Why don't plants just have chlorophyll? I don't know. Tell me. Why should plants also have, in addition to chlorophyll A, why do they also have chlorophyll B and the carotenoids? What's the advantage? Why can they do more photosynthesis? Why can they do more photosynthesis? What do those other pigments do for the plant?
More different lights, more colors. Kind of like if you put it as the accessories.
That's right. What's capital E stand for?
Energy.
Energy. So if you have all these other pigments, they can absorb other colors that chlorophyll A cannot absorb, but these other colors are energy. So you're right. It increases the efficiency of the plant because it's getting more energy from the sun. So Rick is correct. It makes it more efficient in its photosynthesis. Okay, question 4. Okay. Still on question 4. What color of light is the best color of light for photosynthesis? That's a pigment. I want a color.
Green.
Green. White. Why is white light the best color of light for photosynthesis?
Combination of all colors.
And you know what, guys? I think we actually collected some data to support that hypothesis. Check it out real fast on page 73. You should have some data on page 73 where we collected our class data on photosynthesis rate, white light, and then we did green light.
That was with the plant in the water.
That's the plant in the water. It had a light, a big beaker of water that the light was shining through, and then an aquatic plant that would produce oxygen moving water in a tube. Yeah. Okay, number 5. True or false? Most plants carry out photosynthesis only during the day. That's false? That's true? Okay, so what would make it false? Like,
There's no sun during the day.
there's no sun during the day. So most plants carry out photosynthesis only during the day. Is that when the sun is shining? Do you have to have the light to do photosynthesis?
Yep.
Okay, so this is true. Most plants carry out photosynthesis only during the day. That's true. Because you got to have sun. And night is whenever there's no sun. I don't know, you can look up the definition, but I'm really feeling like there's some reference to the sun in the definition of night. Okay, you're with me. True or false? Plants carry out cellular respiration only at night. That is definitely false. When do plants do cellular respiration? All the time. Okay, so cellular respiration makes ATP. Do cells need ATP? When do cells need ATP? All the time. All cells need ATP all the time. So plants are doing cellular respiration every day, every night, all the time. So that's what makes this false. Plants do cell respiration all the time. You do too. All cells do. All the time. Everybody okay on my true-false questions? Number 6. Would you join me on page 74 for number 6? On number 6, why did the phenol red turn yellow when CO2 was added? I'm feeling I need a chemical reaction here. Why did the phenol red turn yellow when CO2 was added? Okay, well, we started with water in a beaker, and then I gave you a straw, and I told you you had to blow in the beaker. What gas did you blow in the beaker? And what did you make in the beaker? It's an acid. Mamadou's right. It's carbonic acid. That was carbonic acid. You guys have that on page 74? Yeah, you made an acid. And so that's why we put the phenol red in there, so we would know when you had loaded it up with acid. Now, then we also talked about this. So phenol red is red when you tell me CO2 is high or CO2 is low? Low. And phenol red is yellow when CO2 is high or CO2 is low? High. So you blew and blew and blew and blew until you got yellow, getting lots of CO2 into the water. And we would know there was a lot of CO2 in the water because the CO2 combined with the water and made carbonic acid. But okay on question number 6, part 1.
So it turned yellow
Because we added CO2. I'm sorry, because we made an acid. Mamadou told us because of the acid. Now, I think we drew the chemical reaction this way on our first day, and then when we came back the next time, I think I did this. Look. What did I just do up here? Yeah, I told you, it could go either way. Okay, go back and forth. This chemical reaction can go back and forth. And so that takes us to my next drawing. Yes, I spent a lot of time on this. Yes. You are impressed. Thank you. Why did the solution turn back to red after being placed in the light? So we left it over in that window, remember? Came back a couple days later. You picked it up and we talked about it. I think you told me it was kind of a pink color, pinky color. Yeah? So why did the solution turn back to red after being placed in the light?
because the plant reduced oxygen.
Nope.
The CO2 from the water. Photosynthesis.
Okay. Okay, what did the plant use? Yeah, the plant used the CO2, absorbed it, and used the CO2. Okay, and so the CO2 level went down. And then I heard somebody say because the plant was doing photosynthesis. So I kind of heard a combination of comments. Okay, now don't get me wrong, the plant did make oxygen. But oxygen doesn't change the pH. So for this particular experiment, we weren't looking at oxygen production. We were looking at CO2 usage. CO2 changes the pH of the water, oxygen doesn't. So in this particular experiment, we were looking at what was happening with CO2. Okay, next question. That's supposed to be a moon.
Because there was no light.
Modern art right there.
That's modern art. Thank you.
To allow photosynthesis.
So I'm going to put no light, no photosynthesis. So when we turned 2 days later, when we took our beakers out of the drawer, they were either yellow or orangey yellow. So we could clearly see that not much photosynthesis had occurred because there was still plenty of CO2 in the water. But okay on that one. Question 7. Why is it important to have autotrophs, heterotrophs, and decomposers in an ecosystem? Well, again, I spent much time on this. I drew an ecosystem. It's very nice, thank you. It's an aquatic ecosystem. Let me point out a few things. Can you guys find the autotroph in my little ecosystem? What? The plant. Can you find the heterotroph? The little goldfish, yes. And yes, they are kind of pooping there. Do you guys see it right here? I have a fish that is pooped. Okay. Yeah, it's pooped. Okay, and then I've got some gravel down at the bottom. If you've ever had an aquarium before, you know you got to put gravel on the bottom. Does anybody know why you need the gravel on the bottom?
Isn't that a habitat for decomposers?
Excellent, Zach. That's my habitat for the bacteria, which are my decomposers. Okay, so why do I need the plant?
So it's cellular respiration.
Provide food for the fishes.
Provide food for the fish. What? Look up there. What else?
Like oxygen.
To make oxygen for the fish. And so I have the plant making oxygen and the plant making the food right here. Okay? Why do I need the fish? To make the carbon dioxide. So the fish is doing cellular respiration. It's making carbon dioxide. And the fish is also urinating, releasing some water. Yeah. Why do I need the bacteria?
To get rid of the nastiness.
Okay, yeah. So the fish pees, urinates. Does that waste product need to be broken down?
Yes.
Yeah, the bacteria can do that. The fish poops, also known as defecation. Does that feces material need to be broken down?
Yes.
Yes. Okay, so the decomposers recycle materials. Yes? So do we need decomposers in ecosystems? Yes, definitely. They're very important. They help recycle material in our environments. Okay, so you guys see the cycle here. We've commented on the cycle before, where oxygen is released and food is released by the plant, and then the animals breathe the oxygen, eat the food, release the CO2 in the water, and it's cycled back over. So you got a nice cycle of materials here between photosynthesis and cellular respiration. Everybody okay on that? Okay, do you notice one more thing in the picture I haven't talked about? It's in the top. The sun. Do you need it?
Yeah.
Sure. It's our energy input into this system. Got to have the sun. Okay, on question number 7. It's just basic ecosystems. Great. Okay, so did anybody need anything repeated on photosynthesis? Anybody? Okay, great. One last comment. This picture that I just did, it's kind of like the picture on page 74, but instead of my aquatic plant and goldfish, there's a cow and some corn. Where's the bacteria in this picture?
Soil.
In the soil. Good. Okay, cellular respiration, page 77. Now, the reactants and products of aerobic and anaerobic cellular respiration. So I've got one up here. Look, I took this from your book. Does anybody know which one this one is? This one's aerobic. What makes it aerobic?
The oxygen gas.
Good. And then I'm missing a number in front of the ATP. 38. Quick question. What is this C6H12O6? What is that? It's a carbohydrate, and most commonly it is glucose. Thank you. Most commonly, most of the time it's glucose. Sometimes it's fructose or galactose, but it's usually glucose. Just want to comment in case you don't know, C6H12O6. That's glucose. And I found this on page 77. This particular chemical reaction I found on page 77. Okay, this next one I found on page 78. You want to turn the page 78. And I think we wrote another one that I have up here. Didn't we write this on page 78?
Yep.
Okay. One of these is alcohol fermentation, and one of these is lactic acid fermentation. Which one is alcohol fermentation? The top one, yes. Everybody check out alcohol fermentation makes a gas. What gas? Carbon dioxide. Whereas lactic acid fermentation does not. Let's find two things these have in common. Can you see two things they have in common?
Glucose is a reactant.
Glucose is a reactant,
And 2 ATP is part of the product.
and 2 ATP is part of the product. I see the similarity there? There's a word that describes these two chemical reactions, and it is anaerobic. The word is anaerobic. What makes these two reactions anaerobic? There's no oxygen as a reactant. Everybody check it out, no oxygen. These are anaerobic. It takes care of question number 1. Look at question number 2, so easy. Both anaerobic and aerobic cellular respiration ordinarily begin with what molecule? Thank you. Who was that? Daniela, very good. Glucose. Okay, you get to choose from all 3 of the chemical reactions. You get this one, and the first one. You get to choose from all 3 chemical reactions to answer the question. In which, number 3, in which is glucose completely broken down?
First one.
It is. It's aerobic. That glucose is completely chopped up all the way down to the basic molecules. And here's how you would know. You'd look at the ATP yield. Yeah. How much ATP does each reaction yield? Well, the one that chops the glucose up completely yields the most. And aerobic cellular respiration yields how many ATP? 38. And the others only yield 2. Because they hardly broke the glucose down at all. Thus, they didn't get much energy out of the glucose. But okay, on just this question, how much ATP does each reaction yield? Everybody's okay on that one? Okay, question number 4 is our fermentation experiment. Would you go over to page 82? Here's where our data is. Okay, first of all, you're going to have to know this was the fermentation experiment. This is where we had to put sugars and yeast in these itty-bitty test tubes. And we had to push them up inside of another test tube and then flip it. And we looked, and we put them over in that incubator so they'd get all warm. And then we went back and got them out, and we looked for gas. And I don't know if you can tell, but I have some gas in these right here. Yeah. We measured gas production. Just put arrows on the gas. You're going to have to know what the gas is. What gas was that? It's CO2. Are you guys saying CO2?
Yeah.
Okay. CO2. This gas is CO2. You got to know what gas this was. What gas? Okay. This was fermentation. So was this aerobic or anaerobic? Anaerobic. This was anaerobic. Squeeze it in. Okay, just glance real quickly back. Page 77, one more, no, 78, I'm sorry, page 78, one more time. Those are your two choices on fermentation. Two chemical reactions, page 78. Which one did we do? The top one, how do you know? Because the bottom one, does it make CO2? Did the bottom one make CO2? No. Did the top one make CO2? Yes. So we were doing alcohol fermentation. Do humans use alcohol fermentation for anything to make money?
I'm sorry, say that again?
Do humans use alcohol fermentation for anything to make money?
Oh, yes.
Oh, yes. So it has commercial value. What can you make using alcohol fermentation? Beer, wine.
Whiskey.
Thank you for not thinking about alcohol. Breads, all the bread. So what makes alcohol fermentation important in making bread? Like why? Why do you need it? The yeast what? The yeast makes the bread rise. So what is the yeast making that makes the bread rise? Carbon dioxide. It makes bubbles in the bread. That makes the bread rise. The bubbles in the bread are from CO2 from the yeast. Everybody knows you got to have the yeast. It's the gas coming from the yeast that makes the bread rise. Everybody's okay with that? Okay, how come when you eat bread you don't get drunk? You'd still eat it.
That'd be something.
That would be something.
2 birds, 1 bread.
Right? To chop a burger, I guess you cook it out of it.
You cook it out. Alcohol evaporates. Yeah, well, alcohol evaporates really easily, doesn't it? And alcohol evaporate? Sure it does. So when you put it in the oven, the heat makes any alcohol that was made, it just turns into gas and floats away. Ma'am?
What was your question?
Why don't we get drunk when we eat bread? Alcohol evaporates, yes.
So you're saying that if someone really wanted to, they would just eat the dough and get drunk?
Yeah, but they would have to be hard up. It's kind of like buying NyQuil. They just have to be really hard up to that. You'd have to eat so much. Yeah, you'd have to eat a lot because it's not making a lot of alcohol. So that would just be silly. Yeah. And expensive. Okay, so I think I answered this question. Did the gas bubble increase or decrease with each? And I think it increased in all 3 of them. Does our data support that? Did we get more gas production in all 3 tubes? What was the purpose of tube number 4, which did not have sugar? Negative control. Let's see, why the yeast used sugar. To make ATP. You know, the yeast doesn't care that it's making CO2 and it doesn't care that it's making alcohol. The yeast wants the ATP. We want the other things, but the yeast wants the ATP. And of course, we already answered the question, what was the gas? And the gas was CO2. So the gas bubble increased with each tube, except tube number 4, I don't think we had much gas production in tube number 4. Is that right? Not much is our negative control. Everybody okay on question number 4? Okay, our aerobic cellular respiration experiment. So our aerobic cellular respiration experiment was where we had a ring stand with a clamp. This is supposed to be my clamp. And in the clamp we put a big test tube with cotton at the bottom, and then some soybeans, a top with a pipette. This is the experiment. We use shaving cream. Do you guys remember that one?
Yep.
And if you don't, just take a quick look with me back on page 79. Those were the 3 tubes that we did. Cotton at the bottom with KOH. And then test tube number 1 had some germinating peas. Test tube number 2 had some dry peas with glass beads. And test tube number 3 just had glass beads. That's the aerobic respiration experiment. Did you guys write down that that one was aerobic? And the reason it was aerobic is we left air in the test tubes. So we left them some oxygen, gave them some oxygen. And of course our cells were the peas. So question number 6 is in relation to that activity. Which direction did you expect the shaving cream in the pipette to move? In or out? In. Why? I heard sucking it in. I'm going to use the word vacuum. Because vacuums suck, right? Like a Dyson, they're really good. I don't know if you have one of those. But anyway, that's not the point. The point is a vacuum, the word vacuum means to pull a suction because you create low pressure here, so it pulls from the high pressure. That's the whole idea of a vacuum. So plants used the oxygen inside the tube, and that created a vacuum, and then that sucked the shaving cream inward. The idea was the more oxygen they're using, the more they will suck. So that's why we were measuring our shaving cream. So we already answered question 7. What gas was taken up by the germinating peas? O2. I have a real quick question. Why did we wrap those tubes in aluminum foil? To stop photosynthesis. You may remember we also wrapped those tubes with aluminum foil, and that was to stop photosynthesis. It's good. Okay, that was exercise number 7 on cellular respiration. You guys okay with that? Yeah, okay. Mitosis. All right, so you need to know what happens in interphase, prophase, metaphase, anaphase, and telophase. I want to describe it, and you just tell me what I'm talking about. So this is a stage of cell division where the chromosomes line up in the middle of the cell.
Metaphase.
It's metaphase. How about this is the stage of mitosis where nuclear membranes reform and either a cleavage furrow or a cell plate also forms.
Telophase.
And what's telophase? So this is the stage in the cell cycle where the cell grows and copies its DNA.
Interphase.
That's interphase. The cell grows and copies its DNA. That's interphase. How about this one? This is a stage in cell division where chromatids move to opposite poles.
Anaphase.
That's anaphase. This is a stage of cell division where nuclear membrane disintegrates and the dispersed chromatin becomes tightly coiled chromosomes.
That's prophase.
That's prophase. And that was all. That is all going to be on our lecture test in a few weeks, where I describe it with words, and then you pick it out. That's going to be lecture type questions. Okay. Oh, sorry. Is it back?
Yeah.
Great. Okay. I found the cell cycle on page 86. So I just took a picture of the one. This is the one actually from your textbook. It's similar, but a little bit different. You find it on page 86. So what I want to do on this cell cycle is draw DNA how it would exist. So I want you to draw in your book. And I'm going to start with G1. I don't know that we're on a number. Yeah, we're on number 1. Know the form of DNA in each stage. That's where I'm at. Thank you for that question. Okay, so DNA can be dispersed or tightly coiled. When it's dispersed, we call it chromatin. When it's tightly coiled, we call it chromosomes. You guys shake your head like, yes, I know these words. Okay. In G1, I'm going to get us started. In G1, it's spread out. You guys see my pink DNA up there?
Yep.
Okay. That's called chromatin. It's all spread out. I'm just drawing one. In S, it's still all spread out, but something happens in S. What happens in S? It replicates. So now it's all spread out, but now it's not single-sided. Now it's double-sided. Everybody see that up there? My double-sided chromatin. Okay, G2 would just be the same. But then something happens in prophase. In prophase, the chromatin that's spread out coils up. So I'm going to take that same thing, but I'm going to coil it up. So now it's not chromatin, now it is chromosomes. Notice it's still double-sided. Can you guys see it? Prophase. So I drew it on prophase. We're going to omit prometaphase. Maybe you've already done that in your book.
Okay, I was just going to say, I think I mistakenly omitted the wrong thing.
No, prometaphase is out. Nobody ever gripes about me omitting things. Not ever. Been teaching 31 years. Nobody said a word. Okay, metaphase. Still double-sided. That's metaphase. Still double-sided. Still chromosome. Double-sided. Tightly coiled. Easy to see. But what about anaphase? What happens in anaphase?
Yeah, it splits.
Split apart. So now it's not double-sided anymore. Now it is single-sided. How about telophase?
It unwraps.
Starts to unwrap. That's right. But it's still single-sided. And then that should complete our cycle. Aren't we back to single-sided still? And Zach's right. Zach's right. It's still unwrapping and be getting long like that again. So if you'll glance up there with me, there's only two phases of mitosis where chromosomes are double-sided. What are they?
Prophase and metaphase.
Protophase and metaphase. And there's two parts of mitosis where the chromosomes are single-sided. That is anaphase and telophase.
So you said that telophase was still like
It's still chromosomes, but they're starting to uncoil. Okay, now I'm going to star S. See my star?
Yep.
And I'm going to star anaphase. Why would I star those two things? I have something in common. Say it again, Michaela. So Michaela said that's where the double becomes a single at S. That's where the single becomes a double at S, and then the opposite happens at anaphase. The double then goes back to being single. So they're undoing each other, if you will. They're opposites, is what Michaela said. She saw the relationship there. They're opposites. S is where the single became double. Anaphase is where the double became single again. So they're just undoing each other. But okay what I did there?
Yep.
And that was all number one. So from number one, it said know the form of DNA in each stage. But real quickly, I just want to make sure you know the difference between chromatin and chromosomes. One of these is spread out, and one of these is tightly coiled. Which one's spread out?
Chromatin.
Good.
chromatin.
Good. And don't forget, it's chromatin during what part of the cycle? Interphase, during interphase. I'm supposed to say interphase. Chromosomes are tightly coiled. And when are they chromosomes? during the cell division, during mitosis. Good. So chromatin is the DNA, and it's all spread out, and that happens during interphase. And chromosomes is when the DNA is tightly coiled, and that happens during mitosis. So I'm just going to back up to our drawing so you can see what we're talking about. The all spread out during interphase, the tightly coiled during mitosis. But okay on that one. Good. Ooh, good pictures. What are these pictures of? Drawings of? Chromosomes. Can you label a chromatid?
One of the appendages, right?
Yep. So I could say it's right there or right there. Those are chromatids. Can you label centromere? The middle, where they're attached. Why did your teacher draw a red one and a blue one about the size, same size and shape?
Yeah, for different.
One from your mom, one from your dad, and we call this pair homologous. What does that mean? The same. You got a chromosome that's real similar from your mom and another one from your dad. Or homologous. They're like partners. Kind of think of them as partners. And one partner came from your mom and one partner came from your dad. These two chromosomes are homologous. We labeled parts on page 86. This was from page 86 at the bottom in your lab manual, page 86. Okay, we have some definitions here. How about I say the definition, you pick which word I'm doing. This is a pair of organelles found in animals, and they're made out of protein. Centrioles, yeah. Plants don't have these. They have the spot called the centrosome, but they don't have the centrioles. That's very good. Okay, these are the proteins that pull chromatids apart. Spindle fibers. Well, this last one will be too easy if I gave you the definition because there's only one left. So you help me. What is cytokinesis?
Isn't that the where it splits the cytoplasm?
Excellent. Division of the cytoplasm. Very good, Zach. Cytokinesis is division of the cytoplasm. You okay? Great. Question 3. Differences between plant and animal mitosis. I'm going to give us an easy one. No centrioles on this side. Centrioles on this side. I made a little table here. Plant is on one side and animals on the other. Plants don't have centrioles and animals do. Okay, can you think of another difference? Okay, who has cleavage furrow? Good. Everybody's fine on that? Okay, great. So I drew these. I'm just getting really good at it too. Yeah, I'm proud of myself. I called 1A and 1B. One of these is an animal. One of these is a plant. Tell me what A is. Plant. So where I have the arrow right here, what is that? Cell plate. And letter B is animal. And where I have the arrow?
Cleavage furrow.
What stage of mitosis would this be?
Telophase.
It would be. This would be telophase. This would be telophase. Why is cleavage furrowing a suitable mechanism for animals but not plants?
Because the cell has got cell wall.
Cell wall's rigid, so it won't furrow in. The cell wall is rigid or too firm, and it won't furrow in. Okay, so number 4 I wrote in here to remind me to tell you that I'm going to test you on mitosis in 3 ways. First, I want to build those pop bead chromosomes and simulate the stages of mitosis on the tabletop, just like you and I practiced with. So page 88 and 89 is a really good page if you want to go back and study the stages of mitosis. And the chromosomes are done with artwork and through the middle of the page. Do you guys remember building? Okay. The second way I'm going to test you is slides, and we mounted a slide of animal mitosis, and we found different stages, and plant mitosis. And so across the top of page 88 and 89 are the animal stages that you can go back and review, and across the bottom of 88 and 89 are the plant. And then lastly, I have these models that are back behind Miriam. An animal is there in the blue, which is the left side for you if you're turning and facing that. And then the plant are these really cool ones that are over on the other side. And if you want to take a look at that, if you haven't, take a look at those on your way out. Feel free to take any pictures that you might want of those models also. I think there's interphase, then prophase, metaphase, anaphase, and telophase for each of those. Interphase is the first one, and then prophase, metaphase, anaphase, telophase. They have a question about that. Great. Those are just pictures. Okay, I think we're on the back. Okay, let's do the same thing. I'm going to do a definition, and you tell me what word it is. This is a cell that has one set of chromosomes. Haploid. So what would diploid mean? So what are homologous chromosomes?
The same.
It's usually 2 of them, right?